Genetic sex provides the initial set of instructions that guide how the brain and body begin to develop, and this process starts at the very earliest stages of life. In most cases, biological sex is determined by the combination of sex chromosomes, XX for females and XY for males. While this difference may seem small, the presence or absence of specific genes on these chromosomes sets off a cascade of developmental events that ultimately influence brain structure and function. One of the most important genes in this process is the SRY gene, which is located on the Y chromosome. When this gene is present and active, it initiates the development of testes in the embryo. These testes begin producing testosterone during a critical window of prenatal development. That early surge of testosterone doesn't just affect the formation of reproductive organs, it also plays a major role in shaping the developing brain. In the absence of the SRY gene, and therefore without that early testosterone surge, the body develops ovaries, and the brain follows a different developmental trajectory. What's important here is that genes themselves are not directly coding for male or female brain traits in a simple way. Instead, they act more like switches that control the hormonal environment in which the brain develops. That hormonal environment is what actually drives many of the differences seen later in life. In other words, genes set the stage, but hormones direct the performance. There are also direct genetic influences beyond hormones. Some genes on the X chromosome are involved in brain development, and because females typically have 2 X chromosomes while males have 1, there can be differences in gene expression levels. Although one X chromosome is largely inactivated in females, some genes escape this inactivation and are expressed from both copies. This can lead to subtle differences in how certain neural processes are regulated, including aspects of cognition and resilience to certain neurological conditions. Additionally, the fact that males have only 1 X chromosome means that any mutation or variation in genes on that chromosome is more likely to have an effect, since there isn't a second copy to compensate. This is one reason why certain neurodevelopmental conditions, such as autism spectrum disorders, are diagnosed more frequently in males. It's not solely due to genetics, but the chromosomal setup creates a different baseline level of vulnerability or protection. Another layer to consider is how genes influence sensitivity to hormones. It's not just about how much testosterone or estrogen is present, but also how responsive the brain is to those hormones. Variations in receptor genes can make certain brain regions more or less sensitive to hormonal signals, amplifying or dampening their effects during development. This adds another level of complexity, where 2 individuals with similar hormone levels might still develop differently based on how their genes regulate hormone reception. The interaction between genes and early development is highly time-sensitive. There are specific windows during which genetic and hormonal signals have a much stronger impact on the brain. If those signals occur at the right time, they can shape the formation of neural circuits in lasting ways. If the timing shifts, the outcomes can differ significantly, even with similar genetic inputs. What emerges from this is that genetic sex is not just a label. It's the starting framework that determines how the brain is exposed to hormones, how it responds to those hormones, and how neural circuits begin to organize from the earliest stages of life. Early hormonal exposure plays a critical role in organizing the brain in ways that persist throughout life. During prenatal development, the brain is highly sensitive to chemical signals, and hormones act as powerful regulators of how neural circuits are formed. In particular, testosterone in males becomes a key driver of this process. Produced by the developing testes, testosterone enters the brain during specific windows and influences how neurons grow, connect, and specialize. One of the more interesting aspects of this process is that testosterone doesn't always act directly in its original form. In many regions of the brain, it is converted into estrogen through an enzyme called aromatase. This locally produced estrogen then binds to receptors and shapes neural development. So while estrogen is often thought of as a female hormone, it actually plays a central role in masculinizing certain aspects of the male brain during early development. This highlights how the same molecules can have very different roles depending on timing, location, and context. These early hormonal effects are referred to as organizational because they establish the foundational wiring of the brain. They influence the size of certain brain regions, the density of connections between neurons, and the sensitivity of those neurons to future hormonal signals. For example, areas involved in reproductive behavior, stress response, and certain types of cognition are all shaped during this period. Once these circuits are organized, they tend to remain relatively stable, forming a baseline that later experiences build upon. In the absence of high levels of testosterone during these critical periods, the brain follows a different developmental pathway. This is often described as a more typically female pattern, although it's important to emphasize that this is not a strict binary process. There is a spectrum of variation depending on hormone levels, receptor sensitivity, and timing. Even small differences in hormone exposure during these windows can lead to noticeable differences in neural organization. Timing is one of the most important factors in this process. There are specific critical periods during which the brain is especially responsive to hormonal signals. If exposure occurs within these windows, it can have long-lasting effects on structure and function. If the same exposure occurs outside of these windows, the impact is much smaller or even negligible. This time-dependent sensitivity is what makes early development such a powerful phase for shaping long-term brain characteristics. These hormonal influences extend to systems that regulate behavior and physiology later in life. Circuits involved in aggression, mating behavior, stress reactivity, and even certain aspects of learning and memory are all affected by early hormone exposure. This doesn't mean that behavior is fixed or predetermined, but it does mean that there are baseline tendencies built into the system from very early on. Another important aspect is that early hormone exposure sets the stage for how the brain will respond to hormones later in life. It influences the distribution and density of hormone receptors in different brain regions. This means that when hormone levels change during puberty or adulthood, the effects are filtered through a system that was already shaped during prenatal development. In this way, early organization and later activation are tightly linked. The result is that early hormonal exposure creates a foundational architecture for the brain, influencing how it is structured, how it functions, and how it responds to future internal and external signals, all of which contribute to the patterns of difference observed between male and female brains. Puberty represents a second major phase of brain development, where hormones don't just activate existing circuits, but actively reshape and refine them. During this period, the body experiences a surge in sex hormones, primarily testosterone in males and estrogen and progesterone in females. And these hormones begin to influence the brain in ways that build on the earlier organizational effects established before birth. What makes puberty unique is that it combines both activation of previously formed circuits and structural remodeling of the brain itself. One of the key processes happening during this time is synaptic pruning. Early in life, the brain forms an excess number of neural connections. This overproduction allows for flexibility and learning, but it's not efficient for long-term function. During adolescence, the brain begins to eliminate weaker or less used connections while strengthening those that are frequently activated. Sex hormones play a role in guiding this pruning process, helping to shape which circuits are maintained and which are removed. This leads to a more efficient and specialized brain. At the same time, there is an increase in myelination, which is the process of insulating nerve fibers to allow faster and more reliable signal transmission. Hormones influence how and where myelination occurs, improving communication between different brain regions. This contributes to better coordination, faster processing speed, and more stable patterns of thought and behavior as individuals transition into adulthood. The prefrontal cortex, which is involved in decision-making, impulse control, and long-term planning, undergoes significant development during puberty. This region matures more slowly than other parts of the brain, and its development is influenced by hormonal changes. At the same time, limbic regions, which are associated with emotion, reward, and motivation, become more active and sensitive. This creates a temporary imbalance where emotional and reward-driven systems are highly active, while regulatory systems are still developing. Hormones contribute to this dynamic by modulating the activity and connectivity of these regions. There are also differences in how male and female brains respond to this hormonal surge. Testosterone tends to enhance circuits related to motivation, competition, and certain types of spatial processing, while estrogen and progesterone influence networks involved in emotional regulation, social cognition, and verbal processing. These are broad patterns and not strict rules, but they reflect how hormones interact with the developing brain to shape tendencies in behavior and cognition. Another important aspect of puberty is that it increases sensitivity to social and environmental input. Hormones amplify the brain's responsiveness to experiences, meaning that learning, stress, and social interactions during this time can have a particularly strong impact. This is why adolescence is often a period of both vulnerability and opportunity. The brain is highly plastic, and hormonal influences make it more responsive to both positive and negative experiences. Dopamine systems, which are involved in reward and motivation, are also affected during puberty. Hormonal changes can increase the sensitivity of these systems, making rewards feel more intense and motivating behavior toward novelty and exploration. This can drive learning and growth, but it can also increase risk-taking if not balanced by developing control systems in the prefrontal cortex. What emerges from this phase is not just a more mature body, but a brain that has been significantly reorganized and optimized for adult function. The hormonal environment of puberty acts as a powerful modulator, refining neural circuits, strengthening important connections, and shaping how the brain processes information, regulates emotion, and responds to the world moving forward. Patterns of brain connectivity refer to how different regions of the brain communicate with each other, and these patterns show consistent differences on average between males and females. These differences are not about one brain being better than the other, but about how information tends to be processed, integrated, and distributed across neural networks. The key idea is that it's not just the size of specific brain areas that matters, but how those areas are connected and coordinated. In general, male brains tend to show stronger connectivity within each hemisphere. This means that regions on the left side of the brain are more tightly connected to other regions on the left, and the same is true for the right side. This type of organization is often associated with more localized processing, where specific tasks are handled within specialized regions. For example, motor coordination, spatial reasoning, and certain types of focused problem solving can benefit from strong within-hemisphere connectivity because they rely on efficient communication within defined circuits. Female brains, on average, tend to show greater connectivity between the two hemispheres. This is largely facilitated by the corpus callosum, the bundle of nerve fibers that connects the left and right sides of the brain. Stronger cross-hemisphere communication allows for more integration of different types of information, such as combining analytical processing with emotional or contextual input. This pattern has been linked, in a general sense, to abilities like language processing, multitasking, and social cognition, where integrating multiple streams of information is beneficial. It's important to understand that these are statistical tendencies observed across large groups, not strict rules that apply to every individual. There is a significant overlap, meaning many males show connectivity patterns typical of the average female profile and vice versa. Brain organization exists on a spectrum, and individual variation is often greater than the average difference between sexes. These connectivity patterns begin to emerge during development and are influenced by both genetic and hormonal factors. Early exposure to hormones helps guide how neural circuits are formed, and later hormonal changes during puberty refine these connections further. Over time, repeated behaviors and experiences strengthen certain pathways, reinforcing these connectivity patterns. This means that while biology sets the initial framework, experience plays a major role in shaping how these networks are used and maintained. Another layer to consider is how these connectivity differences influence cognitive strategies rather than abilities. For example, when solving a problem, two individuals might arrive at the same correct answer but use different neural pathways to get there. One might rely more on spatial processing and focused attention, while the other integrates verbal, emotional, and contextual cues. These differences in strategy can reflect underlying connectivity patterns without implying any difference in intelligence or capability. Functional brain imaging studies have shown that these connectivity patterns also influence how the brain responds to tasks in real time. During certain activities, males may show more localized activation in specific regions, while females may show more distributed activation across multiple regions. This again reflects differences in how networks are organized and recruited rather than differences in performance outcomes. Environmental and behavioral factors can modify connectivity over time. Learning new skills, engaging in complex tasks, and repeated practice can strengthen connections across different regions, regardless of baseline patterns. This highlights the plastic nature of the brain, where connectivity is continuously shaped by use. What these patterns ultimately illustrate is that the brain can be organized in different but equally effective ways, with variations in connectivity influencing how information is processed. Eye hormones continue to influence brain function throughout life, not just during early development or puberty. Unlike the organizational effects that shape the brain's structure during critical periods, hormones in adulthood have what are called activational effects. These are more temporary and reversible, meaning they modulate how the brain functions from moment to moment, depending on current hormonal levels. This creates a dynamic system where the same brain can operate differently across time. In females, one of the most significant sources of hormonal fluctuation is the menstrual cycle. Across the cycle, levels of estrogen and progesterone rise and fall in predictable patterns, and these changes influence brain activity in measurable ways. Estrogen in particular has strong effects on synaptic plasticity, especially in areas like the hippocampus, which is critical for learning and memory. During phases when estrogen is higher, there is often enhanced synaptic connectivity, which can support certain types of cognitive performance, including verbal memory and learning efficiency. Progesterone, which rises after ovulation, has different effects. It tends to have a calming influence on the nervous system by interacting with receptors that regulate inhibitory signaling in the brain. This can affect mood, stress sensitivity, and even sleep patterns. The interaction between estrogen and progesterone creates a shifting internal environment, which can influence how emotions are processed, how stress is experienced, and how cognitive tasks are approached at different points in the cycle. In males, testosterone levels are generally more stable across longer periods, but still show meaningful fluctuations, particularly across the day. Testosterone typically peaks in the morning and gradually declines throughout the day. These fluctuations can influence motivation, energy levels, and certain aspects of cognitive function. Higher testosterone levels are often associated with increased drive, competitiveness, and goal-directed behavior, as well as enhanced performance in tasks involving spatial processing. Testosterone also interacts with neurotransmitter systems, including dopamine, which plays a central role in motivation and reward. This interaction can influence how strongly individuals pursue goals, respond to challenges, and experience reinforcement from success. These effects are not fixed traits but vary depending on current hormone levels, overall health, and environmental context. Another important aspect is how hormones interact with stress systems. Cortisol, the primary stress hormone, does not operate independently. Its effects are shaped by the presence of sex hormones. For example, estrogen can modulate how the brain responds to stress, sometimes enhancing emotional sensitivity, while testosterone can influence how stress is translated into action or suppression of emotional responses. This interplay creates sex-specific patterns in how stress is experienced and managed. Hormonal changes across the lifespan further illustrate their influence. In females, menopause leads to a significant decline in estrogen and progesterone, which can affect memory, mood, and overall brain function. In males, testosterone levels gradually decline with age, which can influence energy, motivation, and cognitive performance. These shifts highlight how ongoing hormonal modulation contributes to changes in brain function over time. What's critical to understand is that these hormonal effects are context-dependent. The same level of a hormone can produce different outcomes depending on factors like sleep, stress, nutrition, and prior experience. This means hormones are not acting in isolation, but are part of a broader regulatory system that continuously adjusts brain function. The result is a brain that is not static, but highly responsive to internal chemical signals, with hormones acting as ongoing modulators that shape cognition, emotion, and behavior in ways that can shift across hours, days, and different stages of life. Differences in stress response and emotional processing between male and female brains emerge from the interaction between neural circuits, hormones, and past experiences. These differences are not absolute, but there are consistent patterns in how the brain tends to react under pressure, how emotions are processed, and how behavior is shaped in response to challenges. These patterns are influenced heavily by the interaction between stress hormones like cortisol and sex hormones such as testosterone, estrogen, and progesterone. When the body encounters stress, it activates the hypothalamic pituitary adrenal axis, leading to the release of cortisol. Cortisol prepares the body to respond by increasing alertness, mobilizing energy, and sharpening focus. However, how this stress signal is processed in the brain can differ depending on hormonal background. In males, testosterone tends to interact with stress systems in a way that biases the brain toward action-oriented responses. This often involves increased engagement of circuits linked to motor output, problem-solving, and immediate decision-making. The response is more likely to be focused on addressing or confronting the stressor directly. In females, estrogen and progesterone influence stress circuits differently. Estrogen, for example, can enhance activity in brain regions involved in emotional awareness and memory, such as the amygdala and hippocampus. This can lead to a more detailed processing of emotional and contextual aspects of a situation. Progesterone, through its calming effects on the nervous system, can modulate how stress is felt and expressed, sometimes promoting a more reflective or internally focused response. Together, these hormonal influences can bias the brain toward processing the emotional and social dimensions of stress more strongly. Another important factor is how the brain integrates stress with social behavior. In many cases, females show greater activation in networks related to social cognition and empathy during stress, which can promote behaviors aimed at seeking support or maintaining social connections. This pattern is sometimes described as a tend and befriend response, in contrast to the more commonly described fight or flight response. In males, stress responses are more often associated with increased focus on the external environment and goal-directed action, though this varies widely depending on the individual and context. The amygdala, a key structure involved in threat detection and emotional processing, also shows differences in activation patterns. Studies suggest that males and females may engage different hemispheres of the amygdala or show different patterns of connectivity with other brain regions during emotional experiences. These differences can influence how emotional memories are formed, how threats are interpreted, and how long emotional responses persist. Chronic stress further amplifies these patterns. Prolonged exposure to elevated cortisol can alter brain structure and function, affecting areas like the prefrontal cortex and hippocampus. Because sex hormones interact with these same regions, the long-term impact of stress can differ between males and females. For example, chronic stress may more strongly affect emotional regulation circuits in one case and decision-making or impulse control in another, depending on the hormonal environment. It's also important to recognize that experience and learning play a major role in shaping stress responses. Repeated exposure to certain types of stress, as well as learned coping strategies, can strengthen or weaken specific neural pathways. This means that while there may be biological tendencies, they are not fixed outcomes. Training, therapy, and deliberate behavioral practices can significantly reshape how the brain responds to stress. What this reveals is that stress and emotional processing are not governed by a single system, but by an integrated network that is continuously influenced by hormones, brain structure, and experience, leading to patterns of response that can differ on average between males and females while remaining highly adaptable at the individual level. Differences in stress response and emotional processing between male and female brains emerge from the interaction between neural circuits, hormones, and past experiences. These differences are not absolute, but there are consistent patterns in how the brain tends to react under pressure, how emotions are processed, and how behavior is shaped in response to challenges. These patterns are influenced heavily by the interaction between stress hormones like cortisol and sex hormones such as testosterone, estrogen, and progesterone. When the body encounters stress, it activates the hypothalamic pituitary adrenal axis, leading to the release of cortisol. Cortisol prepares the body to respond by increasing alertness, mobilizing energy, and sharpening focus. However, how this stress signal is processed in the brain can differ depending on hormonal background. In males, testosterone tends to interact with stress systems in a way that biases the brain toward action-oriented responses. This often involves increased engagement of circuits linked to motor output, problem-solving, and immediate decision-making. The response is more likely to be focused on addressing or confronting the stressor directly. In females, estrogen and progesterone influence stress circuits differently. Estrogen, for example, can enhance activity in brain regions involved in emotional awareness and memory, such as the amygdala and hippocampus. This can lead to a more detailed processing of emotional and contextual aspects of a situation. Progesterone, through its calming effects on the nervous system, can modulate how stress is felt and expressed, sometimes promoting a more reflective or internally focused response. Together, these hormonal influences can bias the brain toward processing the emotional and social dimensions of stress more strongly. Another important factor is how the brain integrates stress with social behavior. In many cases, females show greater activation in networks related to social cognition and empathy during stress, which can promote behaviors aimed at seeking support or maintaining social connections. This pattern is sometimes described as a tend and befriend response, in contrast to the more commonly described fight or flight response. In males, stress responses are more often associated with increased focus on the external environment and goal-directed action, though this varies widely depending on the individual and context. The amygdala, a key structure involved in threat detection and emotional processing, also shows differences in activation patterns. Studies suggest that males and females may engage different hemispheres of the amygdala, or show different patterns of connectivity with other brain regions during emotional experiences. These differences can influence how emotional memories are formed, how threats are interpreted, and how long emotional responses persist. Chronic stress further amplifies these patterns. Prolonged exposure to elevated cortisol can alter brain structure and function, affecting areas like the prefrontal cortex and hippocampus. Because sex hormones interact with these same regions, the long-term impact of stress can differ between males and females. For example, chronic stress may more strongly affect emotional regulation circuits in one case and decision-making or impulse control in another, depending on the hormonal environment. It's also important to recognize that experience and learning play a major role in shaping stress responses. Repeated exposure to certain types of stress, as well as learned coping strategies, can strengthen or weaken specific neural pathways. This means that while there may be biological tendencies, they are not fixed outcomes. Training, therapy, and deliberate behavioral practices can significantly reshape how the brain responds to stress. What this ultimately highlights is that differences in stress and emotional processing are not about fixed limitations, but about tendencies shaped by biology that remain highly adaptable. By understanding how hormones and neural circuits influence these responses, it becomes possible to intentionally train the brain toward better emotional regulation, more effective coping strategies, and greater resilience, regardless of baseline predispositions.